Welcome to this demonstration of how to explore genotype germplasm resources using the Oryza 19K-RGP dataset within the Gramene Oryza gene search interface. In this video, we'll walk through the process of accessing genetic variation data generated from more than 19,000 rice accessions. These variants have been systematically mapped to 5 high-quality, assembled rice genomes. These include the IRGSP Japonica Nippon Bear reference genome, which serves as the foundational coordinate system for much of rice genomics. Alongside it, we incorporate a Basmati genome, ARC10497, chosen to represent the aromatic subgroup. We also include N22, an AUS variety known for its remarkable drought and heat tolerance, making it especially valuable for studies on climate resilience. Rounding out the panel are 2 important indica genomes, IR64, a widely cultivated, mega variety, and Minghui 63, one of the most influential restorer lines used in hybrid rice breeding. As we explore the interface, you'll see how genetic variants are linked to annotated gene models. Whenever a variant overlaps a gene, the system classifies the likely effect of the alternate allele on transcripts or encoded protein products. Particularly important are deleterious protein-altering variants, such as premature stop codons, frameshift mutations, and disrupted splice sites. These predicted loss-of-function alleles are indexed and easily searchable, allowing users to rapidly identify germplasm lines carrying impactful mutations in any gene of interest. In today's example, we'll focus on OSMADS50, a key flowering time gene. By examining variants that affect this gene, we can quickly identify potentially informative accessions, investigate natural functional diversity, and better understand how genetic differences shape important agronomic traits. Let's get started exploring these tools within the Oryza19k.gramene.org website. First, enter OSMADS50 in the Type Ahead search bar and select the matching gene. This defines a filter and displays the gene in the context of an interactive gene family tree. You can expand or collapse branches and explore differences in the multiple sequence alignment. As you can see, there are several rice genomes in which the MADS box transcription factor domain is not present. Clicking on the orthologs button below the tree modifies the search and shows the distribution of the OSMADS50 gene across 28 rice genomes and its orthologs in Lurgia, maize, sorghum, grapevine, and Arabidopsis. Let's scroll down to the Nippon Bear gene and expand the Germplasm tab. Here you can see there are 2 accessions with a frameshift variant. From here, you can access views of all variants in the Ensemble browser, or you can click on the accession link to request seeds from the Germplasm Center. It is possible to limit the search to the set of genomes I mentioned earlier to make it easier to find out if there are other accessions with predicted loss-of-function alleles relative to the corresponding gene model. Now, the search is limited to these 5 genomes. Let's expand the Germplasm tab on each of them. As expected, there are some accessions in common, but there are others that were not detected in the context of the Nippon Bear gene. This search button will trigger a new search for all genes in which the accession has a predicted loss-of-function allele. You can see a variable mapping rate among the 5 genomes. This apparent discrepancy is primarily due to the fact that the genomes have been independently annotated with different pipelines and evidence. We are developing a pan-gene annotation workflow which would provide a uniform set of gene model annotations. Click the trash can icon to clear the search and return to the welcome page.